A very good afternoon to one and all present, respected ma'am and my dear friends. My name is Zainat Lepcha and I am going to be presenting a seminar today on toxicological screening methods, and the topic for my seminar is the history, concepts and importance of toxicity studies in drug development. So today we are going to be studying about the introduction, the concepts of toxicology, the history of toxicology, and its importance. Now we come to the introduction. Now, what is toxicology? So toxicology is basically the study of poisons. It is derived from two Greek words. The first part, toxikon, it means poison, and the other word, logos, it means study. So before proceeding to study about the toxicology, we should understand what toxic or a toxic substance is. So toxic substance is, it can be any chemical, physical agent or substances that have the characteristics of producing an undesirable or adverse health effects. So any physical agents or any chemical substances which has the ability or the capacity to produce any unwanted or adverse effects in an organism, it is known as the toxic substance. Toxicology. Now, what is toxicology? It is defined as the study of adverse effects of xenobiotic. So, xenobiotic is any substance. It's typically a chemical. It is a synthetic chemical that is foreign into the body. So, toxicology is defined as the study of any adverse effects of a synthetic chemical that is foreign to the body. Now we come into the concepts of toxicology. So toxicity can be divided into three types: the acute, the subchronic and the chronic. Now what is acute? So acute toxicity, it involves harmful effects in an organism through a single or short term exposure. So acute toxicity, it can happen within a single time exposure and it requires very short term to show its effect. For example, if a farmer working in the field with the crops, if he's using a pesticide and through some accident, if he gets contact, if the pesticide comes in contact with his skin, then it causes immediate reaction. Basically, it leads to red skin or the swelling of the skin, blisters, burning, or severe pain. So as we can see that pesticide comes under acute toxic substance because it causes harmful effects in a very short time exposure and within a single time. Now we come to the subchronic. Subchronic comes in the middle between the acute and the chronic toxicity. So subchronic toxicity is the ability of a toxic substance to cause effects for more than 1 year. It causes the toxic effect for more than 1 year but less than a lifetime. That's why it comes under the acute and the chronic. Now we come to the chronic. So chronic is the ability of any substance, or it can be a mixture of substance, which causes harmful effects over an extended period of time, usually due to repeated exposure or continuous exposure. So it basically, after this, it may last for a lifetime to the exposed organism. For example, a certain type of acid vapor. So if we inhale it at a concentration over a very long period of time, later what it happens is it causes the loss of our tooth enamel. Eventually, it leads to the extensive tooth decay. So chronic is like for a repeated exposure, and if there's a continuous exposure, so it leads to the chronic toxicity, which may last for a very long time or actually for a lifetime. So this is the difference between the acute, subchronic and the chronic toxicity. Now we come to the specialty areas in toxicology. The target organs and the system. Now these are the various target organs or a specific organ that a toxic substance targets in order to produce its effect. First, we have neurotoxicology. So, neurotoxicity is one form of toxicity in which any type of biological or chemical or physical agents, when it produces an adverse effect on the peripheral and the central nervous system, especially when it affects the structure, it actually affects the structure and function of the central nervous system or the peripheral nervous system. Now we come to genetic toxicology. So, genetic toxicology is the study of the harmful effects of the toxic substances when it damages the genes, either by chemically or physically, any type of agent. So, when it damages the genes, that is the DNA, so if it is not repaired timely and if it is not repaired correctly, it leads to the mutation of the gene. Now we have the reproductive toxicology. So, reproductive toxicology is also the study of the adverse effects of various physical and chemical agents on the male or the female reproductive system. Why? Due to exposure to chemical or physical agents. Now we come to the immunotoxicity. Immunotoxicology, so it is the study of toxicity of foreign substance called, we know, xenobiotics, and their various effects on the immune system of the organism. Now we come to the endocrine toxicology. So endocrine toxicity results with a chemical when a chemical interferes with the synthesis or secretion or transport of hormones, which is necessary for the endocrine function. It results in the loss of the normal tissue function, the development, growth, or the reproduction of the tissues. So this is known as the endocrine toxicology. Now we come to the target species and systems. So target species, we have the aquatic toxicology. So aquatic toxicology is the harmful effects of any type of physical or chemical agents, basically toxic substances that affects the aquatic ecosystem, is known as aquatic toxicology. So it can be from a subcellular level through the individual organisms to the communities, and as a whole, the whole of the aquatic ecosystem, when it is harmed, when it is affected, it is known as the aquatic toxicology. Now we have environmental toxicology. Again, it is the study of any type of physical or chemical agents to the environment. When harm is done, is known as environmental toxicology. Wildlife toxicology, it is the study of the effects of environmental contaminants on the wildlife species. The wildlife that is present, if any type of harm is caused to them due to the toxic substances, any type of physical chemical agents, it is known as the wildlife toxicology. Now we have veterinary toxicology. Veterinary toxicology is a specialty of veterinary medicine. It deals with the study, the diagnosis, and the treatment of effects of natural, either man-made chemicals, forms of energy, and the gases in the animal kingdom. This is known as the veterinary toxicology. Now we have selected responses. For example, teratology. So teratology, it is the scientific study of congenital abnormalities and abnormal formations. For example, I can give example about the thalidomide disaster. So this thalidomide disaster occurred in 1960s. So pregnant mothers, they were given thalidomide was believed to have this mild sedative effect, and it was supposed to treat morning sickness. So it was given to pregnant mothers. So when the pregnant mothers administrated the thalidomide, when the fetus was born, it was born with phocomelia. The uh, phocomelia is the disorder where the limbs are defected. Some fetus were born without limbs, or the limbs were very short. So this led to thalidomide disaster. So it is an example of the teratology, which definition is, it is the scientific study of congenital abnormalities and abnormal formations. Now we have carcinogenesis. So as the name suggests, any type of physical or chemical agents which causes cancer is known as carcinogenesis. Now we come to the applied toxicology. For example, occupational toxicology. So occupational toxicology, it is the application of the principles and methods of toxicology towards chemical and biological hazards encountered at work. For example, a person working in a biological lab or a nuclear lab. So when he encounters this continuous and repeated exposure to chemicals or any type of physical or chemical agents, so it leads to the harmful effects of the toxic substances, so which is known as the occupational toxicology. Now we come to clinical toxicology, the toxic induced diseases and antidotes. So clinical toxicity is the toxic effect of agents whose main aim is to treat or modify or prevent disease states, or the effect of drugs which at one time were intended to be used as such. For example, any type of drug, if we take more than the required dose, it leads to the toxic effects. So clinical toxicology says that it is the toxic effect of agents whose aim is to actually treat or prevent the disease state, or were intended to be used as such before. Now we come to the forensic toxicology. So forensic toxicology is the detection and interpretation of the presence of drugs and other potentially toxic compounds in bodily tissues and fluids. It determines the causes of death. It is used in forensic labs where it determines whether what type of poison was fed to a person. um, so forensic toxicology deals with that. Now we have the regulatory toxicology. Regulatory toxicology, it uses the scientific knowledge to develop regulations and any other type of strategies for reducing or controlling exposure to any type of dangerous chemicals, is known as regulatory toxicology. Now we come to developmental toxicology. So this is a field of study which is devoted to understanding the relationship between the exogenous exposures and the abnormal development, as well as prevention of such exposures, is known as developmental toxicology. For example, new chemicals and its uses. Now we come to the classification of toxicants. Here we have the target organ, hepatotoxin and neurotoxin. As the name suggests, hepatotoxin is any type of toxic substances that causes harm to the liver, is known as hepatotoxin, and the toxic substances that causes harm to the um, structure and function of the working of the peripheral and central nervous system, is known as neurotoxins. Intended use, pesticide or solvent. The source, it can be either natural and synthetic. It is classified into natural and synthetic. And special effect, example, carcinogen, mutagen, endocrine disruptor. Carcinogen, as you all know, it is any toxic substances that causes cancer. um, Mutagen, what is the mutagen? Is it is an agent such as radiation or a chemical substance which causes genetic mutation. So it is further divided into physical state. It can either be gas or solid. Toxicity, it can be extremely toxic or very slightly toxic. Chemical composition, we have heavy metal and organophosphate, and mechanism of action is divided into anticholinergic, inhibitor, and uncoupler. Now we come to the types of toxic response. One is local systemic, and another is immediate and delayed. First is local. So local toxic response is the toxicity that is caused, the effect that is caused only at the site of contact. For example, any type of skin itching substance, for example, something that causes allergy to the skin. If it is comes in contact, it only affects that particular site. For example, the gastrointestinal tract and lungs. Now we have the systemic is the effect is distant from that of the exposure site, it is serious than the local, and it may be fatal at times. For example, the central nervous system, the kidneys and the lungs. So the types of toxic responses can be either local, it can be systemic, or sometimes it can be both as well. Now we have the immediate one. First thing is minutes to hours after a single exposure. Minutes to hours after a single exposure, it leads to the cause, it leads to the effect of the toxic substances. For example, the example of pesticide, as I told you, a farmer, when he is exposed accidentally to the chemical, suddenly it causes redness and swelling as soon as he comes in contact with that, after a few minutes or as soon as possible. So immediate response is seen. Now we have the delayed one. So delayed one is days to years after the exposure. As I told you, there are certain kind of acid vapors at present in certain concentrations. If we inhale it over a very long period of time, it causes the loss of the tooth enamel. So when tooth enamel is lost, eventually what it leads to is it leads to the extensive tooth decay. So that is the delayed reaction, and the toxic response might be immediate, delayed, or sometimes it may be both as well. Now we come to the reversible versus irreversible. So it is largely determined by the tissues that are involved, the length of the exposure, as I told you, the length of the exposure is whether it is short or long, and the magnitude of the toxic. So reversible, we have the rapidly generating tissue. If it is a rapidly generating tissue, for example, the liver, the intestinal mucosa, the blood cells, so these type of tissues, they are regenerated rapidly. So when the toxic effect attacks these type of cells or these type of places, so the toxicity is reversible. But when the toxicity is damages the central nervous system, the carcinogenesis, the mutagenesis, and the teratogenesis, these type of cases is irreversible. Now we come to the three phases of toxicology. The first one is the exposure phase. Then we have the toxicokinetic phase. Under that, we study about the absorption, distribution, metabolism, and excretion of the toxic substance. And we have the toxicodynamic phase. First one is the exposure phase. So the exposure phase encompasses all the processes occurring between various toxicants and the influence on them of environmental factors, for example, light, temperature, humidity, etc. So exposure phase is thus when he comes to in contact with the exposure, it can be either for a short exposure, or for a very long period of time. It is known as the exposure phase. Now we come to the toxicokinetic phase. Now first one here, absorption. As we all know, absorption is the movement of the any type of substance, in this case the toxic substance, from the local site to the systemic circulation. So it can be pulmonary, the gastrointestinal tract, or the percutaneous layer. Now we have the distribution. So the distribution of a substance within the organism, it is a dynamic process. Like it depends on the uptake and elimination rates, as well as the blood flow to the various different tissues and their affinities of the substance. Now we have the metabolism. So it is the disappearance of a substance in the body. So it may involve either excretion from the body or the transformation to other substances, which is not captured by a specific method of measurement. Excretion is the exit of a substance and its biotransformation products from the organism. So it can be in urine or bile or by feces or by sweat. So the kidneys are the most important excretory organ, as we all know. So this is the excretion. And toxicodynamic phase, it is the dynamic interaction of a toxicant with the biological target and the biological effects that it produces. So toxicology has evolved into three professional areas. The first one is the mechanistic toxicology. Now, then we have descriptive toxicology and regulatory toxicology. So mechanistic toxicology, it deals with the identifying and understanding mechanisms associated with toxic effects. So it basically identifies and it understands how the toxic effects works and the mechanism associated with it. Then we have descriptive toxicology. So this descriptive toxicology is a toxicology testing to provide information for safety assessment and regulatory requirement. So this type of toxicology, it basically provides information so that safety testing is done and the regulatory requirement of the toxic substance. Now we have the regulatory toxicology. So it deals with the risk assessment based on the data that is generated by the descriptive as well as the mechanistic toxicity studies. Now we come to the history of the toxicology. So it is believed or it is said that toxicology goes back in the time since the early man. So while they were out for hunting and looking for food, they must have had the knowledge of poisonous substances. Like they were able to differentiate between the good food and the bad food. Like, for example, the venom of the plant extracts that were there in the ancient time. So in the ancient time, poisons such as hemlock, there's a poison called hemlock. it was used in it was used for the Greek punishment. It was also known as the blood of Socrates. It was given to prisoners or any type of punishment. If a person had to go through any punishment, the Greek people, they used to feed hemlock. On 4000 BC, alkaloids, for example, such as morphine, codeine, laudanum, and are all narcotics. So it was identified that morphine, codeine, laudanum are all narcotics. So in 9th century, Dioscorides, he was a Greek physician who classified the poison, and he said that it is divided into the plant poison, the animal poison, and the mineral poison. He also used emetics in treatment. Um, Correct. Now we have Paracelsus. He contributed greatly to the fields of medicine and toxicology, the use of chemicals and minerals in medicine. He was the first, he coined the word zinc. So Paracelsus said that all substances are poisons. So there is not a single substance that is not poison. The right dose differentiates a poison from a remedy. So he was a physician who set the basics of pharmacology, toxicology, therapeutics, and he investigated the dose response relationship as well. So Paracelsus, what he did was he focused on the importance of the toxicon or the primary toxic agent and a single chemical entity. So at that time, this was in contrast to previous schools of thought that included the concept of mixture. So he said he found out that certain illnesses of the body had chemical remedies that could actually cure them. So he is therefore a precursor to the modern day medicine and the use of vitamins and minerals to maintain the health. Matthew Orfila, he is considered to be the modern father of toxicology. So he did the first formal treatment. Experimental toxicology laid down great success to pharmacology in 1813. So what he did was he introduced this Orfila, the modern father of toxicology, he used autopsy material to toxicology so that he was able to provide legal proof of poisoning. So in 1815, Magendia, he detailed the absorption. He did autopsy as well and chemical toxicants and explained the mechanism action of emetine and strychnine and the absorption distribution of these compounds. So he detailed the absorption and distribution of various compounds in the body. Now, when 1850s arrived, the advent of the anesthetics and disinfectants, they began to understand. So during 1900, there was the discovery of radioactive and vitamins. The NIH, or the National Institute of Health, assigned NIDA, the National Institute of Drug Abuse. So FDA assigned US Pesticide Act was established in 1947. 1950s, the Additives of Food, Drugs, Cosmetic Act was passed. So after that, various toxicological journals, professional government and other scientific organisms, they began to broadcast the representation to toxicology. Now we come to the importance of toxicity studies. First one is to establish a dose response curve. A dose response curve is necessary to differentiate between the minimum therapeutic dose and the minimum the maximum therapeutic dose and the minimum toxic dose. So importance of toxicity studies is to establish the dose response curve to ensure the safety of new chemicals, like example the use of pesticides, drugs or food additives before they are registered for general use in industry or doctors clinics. First, the safety needs to be ensured. So toxicity study helps in the ensuring the safety of these new chemicals. To establish the mode of action or mechanism for a toxic effect that may have been seen in other studies. So it basically establishes the mechanism as how it works for a toxic effect if it has been seen in other studies as well. To produce epidemiological studies to explain observations in the population. For example, like if a person is smoking, so the long investigation into the association of smoking with lung is done. So this is also important for the toxicity studies. To validate new methods of testing or uh, investigation, particularly those conducted in vitro. This is mainly in vitro rather than in animals. So validation of new methods of testing and investigation is done by the toxicity studies. The two basic principles guiding toxicity tests in animals: to check the effect of the test substance on laboratory animals and its direct toxic effect in humans. So it checks the effect. The effect of the test substance is first checked on the laboratory animals, and its direct toxic effect on human is checked. Another one is the exposure of laboratory animals to high doses in order to evaluate its possible hazard on human that are exposed to much lower doses. So it is basically the exposure of the animals, the lab animals. First, it is exposed to high doses so that we can evaluate that its future possible hazard on humans that are exposed to much lower doses. Now we have the importance of acute toxicity testing. First one is to identify the target organ of toxicity. So in order to identify like the specific organ where toxicity has occurred, we require the acute toxicity testing. To provide safety measures and monitoring guidelines for workers involved in the development and testing of test substances. So all the workers and all the people that are involved in this toxicity studies, so safety measures are provided and various guidelines monitored these workers so that no harm is done to them. To provide information needed for the dose selection in prolonged toxicity studies. So for prolonged toxicity studies, like the information is provided the need for the dose selection, that info is provided. To generate data containing the adverse effects of a substance on human, animal health, and environment. So it generates the data that has the adverse effect or the unwanted effect of any kind of substance on either human, animal health, and environment, to provide the basis for which other testing programs can be designed, and for academics and regulating purposes. Example, the classification, labeling, and transportation of chemical agents, as well as awareness. Now we come to the subacute toxicity studies. So these kind of studies, it is the dose that is high enough to elicit definite signs of toxicity, but like not too much so that it can kill the animal. Now we come to the chronic toxicity studies. So this study is basically to determine the organs affected and to check whether the drug is potentially carcinogenic or not. So this kind of test, it extends over a long period of time, and it involves very large group of laboratory animals. So this study, what it does is it determines the organs affected, and it determines whether a drug is carcinogenic or not. It is done for a very long period of time, and it is continuous and repeated exposure of the animals is done to the toxic substances. Thank you.